Sara Virginia Weis, que obtuvo su licenciatura en biología en la Universidad de Yale y su doctorado en biología en la Universidad de California en Los Ángeles. Desde 1996 forma parte del profesorado en la Universidad Estatal de Oregon. Actualmente es profesora distinguida de biología integrativa en la Universidad Estatal de Oregon en Estados Unidos. Su grupo de investigación estudia la relación simbiótica entre los corales y sus algas microbianas asociadas. Estas simbiosis son fundamentales para la salud de los ecosistemas de arrecifes de coral y cuando estas relaciones se rompen debido al estrés ambiental como el calentamiento global, todo el ecosistema arrecifal se ve amenazado. Su grupo examina la regulación molecular y celular del huésped coralino y sus algas simbiontes. Este trabajo contribuye al desarrollo de soluciones para la conservación de los arrecifes. Es miembro electo de la Asociación Americana para el Avance de la Ciencia y en 2022 recibió el premio a la eminencia en investigación de la Sociedad Internacional de Arrecifes de Coral. Cuenta con más de 125 publicaciones y ha sido mentora de 20 estudiantes de doctorado y 13 becarios postdoctorales, además de impartir clases a miles de estudiantes de pregrado. Y finalmente, durante 9 años, fue jefa del Departamento de Biología Integrativa durante 9 años. Bueno, gracias.
All right. I really wish I could be speaking to you in Spanish. I have tried to acquire some since I've been here, but certainly not enough to chat with you. I've been lucky enough to be on sabbatical in Anya's lab since January. I have had a fabulous time. I've learned an enormous amount about what her group is doing and the impact that they all have on reef restoration and conservation. And it's so very different than what I do, and you'll see. And so it's just been very refreshing. I'll also say that ruins have been visited, cenotes have been swum in. So I've had plenty of off time as well, and so have fully enjoyed being in the Yucatan for this time. So today I'm gonna take you into a direction that's quite different than the kinds of questions that Anya's group asks about corals. And I'm gonna focus really on, okay, so the advancing isn't working. Let's try it here. Okay, there we go. Focusing on cell biology. And so I'm gonna talk in sort of 2 areas today. The first is in discovery and talking about the regulation of coral algal symbiosis. And the 2 main areas that my group has focused on through the years is the role of host innate immunity in symbiosis and host symbiont biomass regulation. So how the partners manage to grow together in a coordinated fashion. But I'll also just finish with a short section talking about my learning to look forward and seeking solutions to coral reef decline in my own way as a cell biologist and developing the Aiptasia model system to accelerate discovery and generation of solutions. So this is the way our beloved reefs used to look. This is a photo that I took back in 2007 on Heron Island on the GBR. And of course, there are 10,000 species of cnidarians, and many thousands of those are anthozoans that are the reef builders. And they harbor in their tissues, going a little fast, there we go. Let's go back there. Okay, They harbor in their tissues, cnidarians, we know now, are essentially microbial ecosystems, and they harbor symbionts from all three domains of life. They, of course, today I'm going to be talking about the dinoflagellates, but there are also fungi and some other eukaryotes in host cnidarians that are over here in the animals. But there are also abundant bacteria and archaea. And so it's a microbial ecosystem, all parts of which are essential. But today I'm going to focus on dinoflagellates and their host cnidarians. And here's just an image of a Fungioscoteria larva, this very small larvae in this species, that we then labeled with dye that was able to show you this lipstick of bacteria and then the algae inside. So it gives you a sense of just how complex the holobiont is. So if we look at the anatomy of cnidarian dinoflagellate associations and we look up close at a coral polyp, and again, this is all stuff you all know, but just to get us all in the picture, we have these tentacles, the outside of which are non-symbiotic, but inside of the gastrodermal tissue, that in the inner layer of the animal, are where the algae reside. And the algae take up residence in a symbiosome. So that's an intracellular, very intimate association, whereby the association is based around nutritional exchange and regulation, where the host is controlling the nutrients that enter the algae, and the algae in turn are providing huge amounts of photosynthate, so reduced organic carbon, to the host. And just to give you a sense of a cell biologist's look, this, yeah, my pointer is dying. This, it's dead. This is not a correct diagram, because when you look at the algae, the host cells themselves, when they're caused to fall apart from one another, this is what you see. So this is one host cell with maybe 4 or 5 algae in it. And there's a little nucleus, right? So it's a very intimate environment where the host material the host volume is almost entirely taken up by the symbionts that are inside of them. So that results in some challenges in imaging when you're trying to see what's going on with fancy microscopic techniques, but ways to get around it. So the diversity of symbiotic algae, I think many of you know about. We know now that they're an incredibly diverse group of dinoflagellates. Their diversity is roughly equivalent of a lemur to ape phylogenetic tree, with the ancestral groups being down here named Symbiodinium, the old-fashioned name. But now this whole family of Symbiodiniaceae has at least 9 genera. Actually, there are more and more coming. Like this now is a genus. I can't remember the name of it. And most of the corals in the Caribbean have a Symbiodinium species in them. And most of the soft corals in the Caribbean have a Breviolum species in them. So there's this huge diversity. And what's interesting about that from a solution standpoint is that this host symbiont diversity is, the specificity is complex and dynamic. So it's not cast in stone. The phylogenetic trees of cnidarians and algae don't align. It's more like spaghetti, but there is an ability to potentially work with that specificity as a solution to coral decline. And so, gosh, delay. A question that's being asked by lots of groups, not my group, is can specificity be altered to breed or engineer resilient corals? So there are species of corals or algae, for example, that are quite capable of managing higher temperatures. And so a big question is, can one engineer a coral that has those algae that would better able to ride out some of the stresses that corals are seeing globally? So my group, I've worked on corals through the years, but nowadays my group almost exclusively studies a model system. And I'm going to talk a lot about model systems over the next half an hour or so, but I want to introduce you here to one that I'm sure most of you are familiar with, which is Aiptasia, which has now been renamed, which is just an outrage. It's now Aiptasia diaphana, that's the official name, but us Aiptasia folks still call it Aiptasia. So we just took the italics away, okay? So Aiptasia is a fantastic model because it grows so well in the laboratory. It's clonal, we'll talk about that. You can have sexual and asexual reproduction. You can bleach it experimentally so that you can maintain non-symbiotic animals for long periods of time and reintroduce algae. There's a completed genome now, and the symbionts are although there are several different symbionts, the two main symbionts that are harbored by Aiptasia now have completed genomes and are culturable in the lab. And so there's incredible power that in many cases with corals we don't have. For example, the Symbiodinium phytae that's in many of the species in the Caribbean is not yet culturable in the lab. And so in Aiptasia, you just have so much power, and that's really what we spend our time really focusing on is model systems. And I want to say just from the beginning that my joke used to be, all I do is collate and staple. In other words, and see, the young people aren't even laughing because they don't, nobody prints anything out anymore. But my job in the lab is definitely not to do the experiments anymore. And so I always put this slide up at the beginning to point out that the work that I talk about and that I am a success because of is because of all the people that have been in my lab through the years. And you'll notice that all of the youthful faces and then Virginia's face, oh, look, I have brown hair. It just slowly gets a little grayer anyway. And guess when that was, right? So anyway, it's just, I'm super proud and have been enjoyed mentoring all these lovely people through the years, and they've done amazing work. So, we have big questions in the regulation of symbiosis, many of which have been around for years, like 50 years, and many of which still haven't been answered. And I'm going to talk about a few of these today, but I'm going to kind of break it down. And I'm going to start with immunity. And for this part of my talk, this is probably, if we looked at all the things that the Weis Lab has done, this is where we've spent the most time. And this is where I'm going to show no data today. I'm going to show data over here, but I'm not going to show any data here, but I'm going to give you sort of a meta level view of what we understand about immunity and its role in symbiosis and in bleaching. So the kinds of questions you can ask are, what cellular signals are involved in host symbiont recognition when partners first come together? Does the host mount an immune response that is modulated by the symbiont? How does signaling and immunity change during symbiosis dysfunction and collapse? So those are the kinds of questions my group's been studying for years. And most of you know that the majority of corals start off life in a non-symbiotic state, and they have to acquire their algae either as larvae or as juvenile polyps. And you can do this experimentally in the lab. This is actually a set of images from a coral from Fungus scuteria that we worked on for years, where you start off with a larva. You can grind up a little bit of brine shrimp and you add the algae, you grind up an adult, and you add the algae. And the larvae, they go crazy. They drop down to the bottom of the dish. They trail out this mucus. They swim around. You can see that this is a scanning electron micrograph. The algae get trapped in this mucus and down the toilet they go, and you end up with a symbiotic larva. And you can capture it at a cellular level. So this is EM now, electron microscopy. And host cells that are lining the gastroderm, they're nutritive phagocytes. So their whole job is to eat. And you can see this host cell reaching up with its pseudopods to engulf an alga, okay? And that's the way symbiosis begins. And it's remarkably reminiscent of what happens with so-called professional phagocytes in, for example, the human immune system, like this macrophage that's engulfing yeast, pseudopods coming up, swallowing, and destroying, in this case, destroying their yeast. But in the case of corals, the algae persist. They're taken up, they take up residence in that gastroderm, and they persist. And so one of the big questions that we've asked through the years is, what are the processes that lead to engulfment and persistence? And the reason why this is now so interesting, so when I first started asking these questions many moons ago, there were no genomes. There was not a genome to be seen. I remember having coffee with a friend. She was a cell biologist, and she said, isn't this Human Genome Project stupid? I'm like, yeah, what a waste of time, right? So I've been around a while. So now we know that there's so much value in comparing genomes, and we know now that the cnidarian genome is remarkably similar to human genomes, to vertebrate, to mammalian genomes. And this is definitely the case with immunity. And innate immunity is an incredibly highly conserved ancient set of pathways. And so if we look at it just in a generalized professional phagocyte, you have a microbe that has these decorations, these microbe-associated molecular patterns that are on the outside of the algae. They can't hide them necessarily. And these phagocytes have pattern recognition receptors that have evolved to be able to detect the presence of these MAMPs. And once that MAMP-PRR interaction happens, the phagocyte goes, alert, and it launches a variety of pathways through these signaling cascades to do a variety of things, depending on what it is and what kind of cell it is, either killing the algae in the phagosome, killing the algae with a killing mechanism that spews toxins, starting an inflammatory response in other cells nearby, killing itself to remove the environment, killing the algae by encirculating in a membrane. Well, we now know that all of these things occur in the genome of cnidarians. They're all there, all the same stuff. And there's a lot of detail in each of these steps. So lots of names for MAMPs and PRRs and signaling cascades. But we know now that the host cell interacts with symbiodiniaceae through an innate immune response. And what's cool about mutualism is that rather than the symbiodiniaceae getting destroyed, if it's the right kind, it persists in that symbiosome, so that membrane becomes a symbiosome. And these pathways are stopped. So it's an arrested phagosome is what a symbiosome is. And these are the processes that the Weis Lab has studied for years now. So these are all my peeps through the years, and various studies that we've published on these different processes. Two of my current students, June Chai and Olivia Burley, they're each studying, June is studying the sphingosine rheostat, which is a cell proliferation mechanism that drives the host towards proliferation. Olivia is studying TGF-beta, which is a signaling cascade that promotes tolerance in the host. So tells the host, hey, it's okay. This is the right symbiont. So, these are the things that my group has studied for years. And then, of course, bleaching, dysbiosis and bleaching. It's essentially what we know now is that it is innate immune mechanisms re-engaged. And depending on what insult, it could be one or more of these things being re-engaged as a function of stress. okay? And again, lots of studies by not just me, lots of folks really getting a pretty good understanding of what's going on here. But I want to pause here and talk about stress. What is this stress? Okay, what is this stress? Well, we know that elevated temperature is the sort of environmental thing, but what at the physiological level is that stress? Well, there's for years and years, there's been a very strong set of research that talks about the oxidative stress in symbionts driving symbiosis collapse in the host, whereby the algae gets stressed. The Achilles heel of the algae is the Z scheme, which is the photosynthetic apparatus. It becomes destabilized. Oxidative stress in the algae causes damage in the algae. Reactive oxygen species start to leak into the host, and the host gets sick or is toxic and launches an immune response, which I'll talk about in a minute. But there's another very interesting and likely co-occurring phenomenon that has only been really talked about actively for a decade or so, on this concept of metabolic dysregulation, where heat stress causes a breakdown in nutritional regulation, whereby the host starts dumping nutrients to the alga. The alga, who has been slowly growing because it's been controlled, its growth has been controlled, is suddenly like, oh, nutrients? Well, I'm going to start growing. And so then it stops passing its photosynthesis to the host. The host becomes starved, and this drives collapse. Either or both ways, the end result is an immune response. The host says, the invisibility cloak is off this thing. Get this thing out of me. I want it out, no matter what. And so there are a variety of ways by which the host can remove algae. And again, not necessary to understand the ins and outs of what are important, but it can be destroying the algae inside, exocytosing the algae, removing the environment for the algae either by detaching or killing itself, or just dying. And of course, we all know that differential hosts are differentially susceptible to bleaching, depending on species and environment. And it's that, there's a lot of interest in understanding, is there a way to mitigate bleaching by driving resilience somehow? I want to point out this paper. This is a review paper that I was part of last year. That is it's an absolute tome. It is a masterpiece written by Josh Helgo, who's a student of Mauricio Rodriguez-Lenidis. That is really encyclopedic. It has these spectacular drawings of the processes in bleaching. So if you would like the newest, latest mechanisms that are driving bleaching, this is the place to go. It's very long, it has beautiful figures, but it's something it's something to keep in your back pocket for sure. So with finishing up this section, symbiosis regulation via immunity. In health, we have a healthy interpartner communication. The algae are avoiding immune destruction. The immune system is balanced between tolerating and resisting a negative invader and tolerating a positive one. And there's a dynamic homeostasis. And in sickness, we have a communication breakdown. We get holobiont stress. That immune teeter-totter goes to resistance. And the corals lose their algae by mounting an immune response that results in dysbiosis. And so those are the processes that I spent a lot of time working on for the last 27 years. Okay, moving on to host-symbiont biomass regulation. That's a mouthful. Like, what am I talking about? So let's just think about, let's get at this by thinking about corals a little bit and just sort of everyday things that they do that we don't necessarily think too much about. But Here we have the symbiotic larva, Funguscuteria larva, and we have a few algae in there. And the question is, how does the symbiont population keep pace with host growth? So that little larva is going to settle, it's going to polyp, and then it's going to grow and grow and grow. And if this population doesn't grow fast enough, then it'll get lost, okay? Here's another example. An acroprid growing on the reef. You'll notice that the tips are white. The growing tips are white. They don't have any algae. So how do the symbionts invade new host tissues? Do they get exocytosis? Do they move through the gastric cavity and they invade, or do they divide together? Or, you know, what's going on? These questions are remarkably hard to get at. Think about that host cell size. Think about how hard corals are in many ways. And so this is where model systems are incredibly valuable. And Aiptasia has a very convenient life history. It has asexual reproduction. This is why it's an aquarium pest, because it grows so well without having sex. It leaves little bits of its foot behind. So here's an Aiptasia, and it's left little bits, little pedal lacerates that grow up into little primary polyps, and then they eventually become adults. And transmission of the symbionts is vertical, okay? But you can also have Aiptasia spawning, and I'll talk more about that later, but they spawn, they're gonochoric, so they spawn eggs and sperm, different individuals. They form little larvae, and you can, they can either become colonized as a little settled polyp or as a little planula. So here's a little planula larva with algae in it, and then they grow up. And I mean, to say that these are convenient, it's an understatement. They grow so well in the lab. They're actually quite hard to kill, that you can really make progress in a way that is hard to do on corals. And then also keep in mind that you can render them aposymbiotic. So you can start with a non-symbiotic adult and introduce algae and ask questions about onset of symbiosis. So there are a variety of questions you can ask about host symbiont biomass regulation. I don't really have time to talk about all the ones I want to talk about, but I'm going to talk about, and I have these at the end if anybody wants to know, but I'm going to talk about these two. How are symbiont populations regulated during onset and maintenance of the symbiosis? And how does host nutritional state affect symbiotic host development and growth? So this is a brand new paper by a group of us, but Lucy Gorman from Simon Davies' lab and Trevor Tivey from my lab, that literally came out like 3 weeks ago. I'm super excited about this paper. And this is the question that they asked. How are symbiont populations regulated during onset and maintenance of symbiosis? And I'll explain the experiments they did, but I want to just show you how the pattern of the algae invading the host in the experiment. So this was all stuff that Lucy did. So she started with aposymbiotic adults and she added algae. And she did a variety of species, but I'm not going to talk about that. I'm only going to focus on the native symbiont Breviolum minutum. And you can see that, and she followed them for 1.5 years. Oh my goodness. During COVID, so she was like sleeping in the lab, like crazy stuff. This is all in New Zealand. And she also had long-term, these are just animals that are symbiotic. And you can see that these gray bars are the Breviolum. There's a huge shooting up of algal growth that then plunges down and becomes stable. And that pattern is very, very important to understand. So algal cell populations increase rapidly, followed by a strong correction to a lower state. And the question is, what cellular mechanisms drive this pattern? And this pattern has been observed repeatedly. This is just her data. And what Lucy did is she measured 4 things simultaneously in all of her samples. She measured cell cycle arrest in the symbiont. She did this with flow cytometry. So if this is a depiction of the symbiont cell cycle, when it's growing very, very slowly, all the cells get stuck in the G1 phase. That's the growth 1 phase. They can't replicate. Okay, they don't replicate, and you can figure out this on flow cytometry. So she measured cell cycle. She measured apoptosis by looking at caspase activity. That's the enzyme that drives apoptosis. She looked at enzyme activity, whether or not host cells were killing themselves to remove the environment from the algae. She looked at autophagy, which is this surrounding an alga with a membrane and destroying it. She looked at that by measuring Westerns, so antibodies, using anti-LC3. And then she looked at expulsion via exocytosis by counting the number of algae that were in the dish, that were released in the dish. And she did this for 1.5 years. It's insane. And she got these beautiful data. And, I could spend an hour on this slide, but I'll just try to summarize briefly. She basically showed that with the cell cycle arrest measurements, at week 1, the algae are growing. So most of them, many of them are not in G1, they're in the growth phases. But by the end, the long term in the 78, almost all, this is 100 up here, almost all of the algae are stuck in G1, so they're not growing. So they have stopped growing by week 78 and long term. In contrast, apoptosis starts off strong. Let's just look at the pink bars, which are symbiotic. Let's look at the gray bars, which are symbiotic. It starts off strong, so there is some host cell death, but there's a crash at 4 weeks, followed by a recovery. So there's a huge depression in apoptosis at 4 weeks. Autophagy, there's some throughout. There's not a whole lot, but there's some throughout. And with expelled algae, if we look at expelled cells per microgram of host protein, so indexing it to host protein, again, there's a big crash in expulsion early on, but then it becomes important later. So that was Lucy doing crazy stuff during COVID. Think about this experiment. You have to get up that many times a day for all those weeks. Oh my goodness. And then along comes Trevor, who's a modeler. He's mathy. And he basically took these data and he asked, what combination of these 4 things results in the correct pattern that we observe of that big spike and then a correction? How many, is it 2% cell cycle? Is it 50% apoptosis? So he did these hundreds of models and he came up with a beautiful answer. And these are the data that he showed. So the green is the spike, the data themselves, the observed result, with the algal cell population spiking and then crashing and going to a stable state. And this final model that was the closest representation of that was basically for the first 9 weeks, 76% of this is responsible by cell cycle arrest, 8% from apoptosis, 3% from autophagy, and 13% from expulsion. After 9 weeks, greater than 99% of the effect was from cell cycle arrest alone. And so there are some very key points to learn here is that during onset of symbiosis, all of this cell biology is happening. All of this regulation is happening. But when you get past 9 weeks, the host is controlling cell cycle, algal cell cycle, to become the dominant maintenance throughout. 109. Okay. Next question, how does host nutritional state affect symbiotic host development and growth? This is work that was done by a former postdoc of mine. Again, when we look at Aiptasia and we can look at pedal laceration, they then form these little polyps as they go. And Sam became very interested in the question of nutrition and the role of nutrition. And he hypothesized that the presence of symbionts in pedal lacerates would boost partnership growth and survival during starvation. That, you know, if you're going to starve, better to have your symbionts there to be tiding you over when things are rough. And so he did a pretty simple experiment where he just had 3 different treatments: aposymbiotic, inoculated, meaning he added algae partway through, and then symbiotic. And he made little pedal lacerates by cutting the foot. And he either fed, he had a fed treatment, fed with little rotifers, or starved them for the entire 60 days. And then he followed just host survival, growth, and algal population size in the animal. And shocking results. This is why science is so awesome. Like, it's so obvious. You have symbiosis. Symbiosis is always good. You know, you always want your symbionts around. They're always going to bail you out. Totally wrong. Totally wrong. Okay. He showed that if you starve them, okay, so let's look at this. We've got the filled circles are fed and the open circles are starved. The aposymbiotic is gray, inoculated is kind of brown, and the symbiotic is green. So if you're fed, no matter who you are, you do okay. Even the apos way up here are doing just fine. They all survive. But if you have algae, you're toast. You start to die really quickly, like before 20 days. If you get your algae later, you make it for a little while longer. But by the end, almost all the symbiotic animals were dead. Like total shock. Growth also dramatically lower in symbiotic animals. Again, if you're fed, even as an aposymbiotic animal, you do okay. The best, the animals that do the best are the symbiotic animals. But an aposymbiotic animal, you do okay. If you're an aposymbiotic and you get starved, you just hang in there. You don't croak, you just like don't grow. You're like, I'm waiting for some food. But if you're symbiotic, you're toast. They just don't grow. They got smaller and smaller as the experiment went on. They became little balls. And the algae went nuts. So the algae in the fed guys, they behaved themselves. They stayed at a fairly stable place. Because remember, these are vertically transmitted. So the algal population comes with the host tissue. But if they are starved, the algal population soars. So what does that look like? That looks like a dysregulation, okay? A massive, totally the opposite of what we were expecting. So starvation causes nutritional dysregulation, homeostasis breakdown, and the algae move towards parasitism. So if we go back to this slide, coral bleaching, what is the stressor? And we focus on this, which is this metabolic dysregulation. Okay, you can do it. Okay, of course. There we go. This is now looking kind of familiar, where we have symbiosis dysregulation. We have starvation causing a change in nutritional regulation. Probably the host is catabolizing its tissues, starting to send a bunch of nutrients over the algae. The algae are like, oh, this is nice. Maybe I'll just stop feeding my host. And the symbiosis homeostasis is lost. But what's interesting is the host doesn't seem to be able to figure it out. It doesn't mount an immune response. So is the immune system immature in a juvenile? It seems to be related to developmental state. Is there a similar effect in F1s in the reproductive animals? Well, we can check that out. Does this make us think carefully about the size of our fragments that folks who are doing restoration are doing? You know, is there a size that's too small? So this was a totally surprising result, but very cool. So when we think about symbiosis regulation and host symbiont biomass in health, we have nutrient cycling in partners that's regulated. Host size and algal population size are balanced, and there's a dynamic homeostasis. But during sickness, We have nutritional dysregulation. The symbionts move towards parasitism, and homeostasis is lost. Okay, so for this last little bit of my talk, I'm going to talk about something different, which is learning to look forward and sort of managing that as a cell biologist. Coral reef decline, all of you are living this, being here right in Mexico. But, you know, in Oregon, I'm a long way away from it. I study cell biology. I'm not a field person, never have been, not an ecologist, none of that stuff. It really resulted in an identity crisis for me and for others. And, you know, what should we be doing to help with the crisis that you all are on the ground directly impacting? And I had been working on this a little bit through time, but I really became very focused on trying to help the field understand the value of model systems by promoting, facilitating, and building Aiptasia across the community. And, you know, with that knowledge and symbiosis, we can directly impact things like enhancing resilience, things like, you know, engineering solutions. And so I just want to give you a sense of just how far this effort is now. The first time that we got together as a group, it was actually John Pringle and me and Simon Davey. We convened a workshop on Heron Islands, and we tried to convince people that model systems were important for corals. And they were skeptical, including Madeline Van Oppen, who drank the Kool-Aid ultimately, and Oren Levy. They were both like, you know what? Corals are the only way to go. Model systems have no value. But we also asked a bunch of pathologists and parasitologists to come to try to give us a sense of how model systems are a playground for figuring out what might be. And it really started to take off. And this is a current rough version of the people who were involved across the globe in Aiptasia in a major way or even in a minor way. And they're people who've been doing it for years. They're people who have recently joined the cause. There are quite a few undergrad institutions in the US that use Aiptasia in their studies with their undergrads. But again, the emphasis is on cooperativity and collaboration and a mission of really driving the understanding of symbiosis forward. And so we've developed, I was awarded a big NSF grant a number of years ago where we helped develop the model systems. We convened a workshop. We have a protocols.io site. Very significantly, we have a course source site for college teachers or university professors who want to start to use Aiptasia in the classroom. It's an incredibly powerful thing. We have a website. Beautiful Aiptasia even made the cover of Lab Animal. That was my moment of fame. But I really feel that as a community, we've come a long way. We've together made sequenced genomes. We share animals. It's important, and we're accelerating the cause of science. But I want to finish with a thank you to Anya. I want to say Anya and I we are in the same academic family. And my advisor, Len Muscatine, was also the advisor of Bob Trench. And so here I am 25 years ago, and here's Anya. And these are our siblings. Do you know who that is? I have no idea who that is. Yeah. There's also some poor person behind Len. I don't know who she is either, but I don't know who he is. Get off there. But all of these folks, Anya and I all know quite well. And so I'm the auntie and Anya's the niece. As is Roberto, who's right here. And Anya and I reconnected in 2020 in Costa Rica. I had become involved in the Coral Restoration Consortium I chaired a working group on cryopreservation, and Anya's been involved with CRC for many years now. But we reconnected there, and I thought, that's the place for a sabbatical. And so here we are last week, and it's just been absolutely wonderful, Anya. I've just had such a great time. But there's a little science here, and that is that at home in the Weis Lab, we have monthly spawning of Aiptasia. Every month you can entrain them to blue light, and they'll spawn millions of animals. So we keep our animals in these little dishes. We keep them at night when they're about to spawn. You can just kind of see there's an animal that's like this. Oh, it's about to dump its spawn. And then this is the outcome. Millions of eggs, little embryos, and then of course a symbiotic larva. And so Anya thought, aha, perhaps this is something that we could do at UNAM to try to accelerate her group study of cryopreservation and innovation of cryopreservation. And so Franja and Vicky have set up this beautiful tank with these giant animals that are thinking about spawning, haven't done it yet. But the goal here is to accelerate innovation in coral cryopreservation and allow for training on material year round. And so that's Aiptasia rules the world is all I can say. So I can just say that now, hopefully, Aiptasia will be contributing to repositories and that we can add a little dot at UNAM. And so I'll stop there. This is just to say that this is my current Weis group. I didn't even talk about any of their work, which is terrible, but next time I will. I have other collaborators who I've worked with for years. And of course, Simon and I have been collaborating for decades now through the Marsden Fund, which is a New Zealand fund. That's where we got our money for the nutrition paper we talked about. And I would just say, you know, we can't give up on reefs. I know you guys haven't, but not even the wise know all ends, according to Gandalf. So, muchas gracias.
Gracias. ¿Preguntas? Quedan unos minutos para preguntas aquí en la sala. Aquí hay una pregunta de la doctora Ania.
Fantastic talk, thank you. How do you think the very first symbiosis initiated? I mean, given the complexity of the onset of symbiosis, now that they know each other, how do you think it started?
You mean in evolutionary time?
In evolutionary terms.
Yeah. So cnidarians, they have primitive is an insult, so I won't say primitive, but they have very ancestral feeding where most animals secrete enzymes into a gut and digest things extracellularly. Cnidarians do intracellular digestion. And so to me, the most parsimonious explanation is that algae were brought in as food and that somehow over evolutionary time, they managed to fool the host, right? And they started that, all is well. I'm cloaking in place. Don't eat me. I'm you. And that all went great until bleaching came along, right? Where the whole thing breaks down. So that's what I think. And, you know, it's a great question is it, were all cnidarians once symbiotic and they've been lost? And so that's why it's spotty? There's no way. It's got to be independent evolution. Yeah.
¿Alguna otra pregunta en la sala? Denise.
No tenemos preguntas en línea. Recuerden que pueden levantar la mano o escribirla en el chat, pero no, todavía
Gracias,
no hay.
Denise. Aquí tenemos una del doctor Marco.
Hi, Virginia.
Hi.
I have a question about the cell cycle arrest. Is it known? How is it induced?
No. And the problem with, so that's the next obvious question, right? With that study. The challenge with cell cycles is that they're so pleiotropic. So in other words, there's so many things that feed into cell cycle control that it's really hard to try to isolate one best. I would love ideas for that because I think that's that's the, because we are starting to get some CRISPR and things where we can really manipulate things. That is really the $64,000 question, right? It's very hard to get at. Great question.
Okay, another question I have is the experiments of the shocking results were done on Aiptasia, right?
Yes.
Okay, how different would it be for corals, taking into account that Aiptasia can survive bleach,
Yes.
but corals cannot?
Yes. So I'll slightly sidestep your question, because one thing I took out to shorten the talk a little bit is what's even more shocking about that result is it's the opposite in adult Aiptasia. So in adult Aiptasia, and this is not studies from my group, in adult Aiptasia, you can starve them for 1 year. So you take an animal, I might still have the slide. Well, anyway, you can take an animal and not feed it for a whole year, and it shrinks and shrinks and shrinks and shrinks, but it stays symbiotic. And so that homeostasis is maintained. And so to me, the shock is why in adults is homeostasis maintained and the algae remain mutualistic, but in juveniles, it is not. And so in corals, there is recent work, the Wiedemann paper, the Nature paper, that showed when they starved corals for like a year, 9 species of corals, they eat their algae. So again, homeostasis is maintained. There are regulatory mechanisms in place. But I think what's novel about Sam's work is that it's that juvenile phase where all bets are off. And so when you have regeneration, which is what a lacerate is, there's evidence from the literature that those regenerating tissues are immunologically naive. And so I think, and this is testable, I think that the reason why the symbiosis fails is that the innate immune system has lost its maturity and is reset and it's a baby again. So anyway.
Okay.
Yeah.
Lots more questions.
Thank you. Gracias por asistir.
Gracias, gracias.